So in this one we're going to be talking about ion channel coupled receptors. There are around about 4 forms of these types of ion channels. You've got ligand gated, where a ligand needs to be on the outside. You've got ligand gated where the ligand needs to be on the inside, so they're sort of the same thing but a little bit different. You've got voltage gated, which means it only opens at a certain voltage. And you've got stress gated, meaning it only opens under stress. And then there's types of ones where they aren't actually receptors and they're just sort of like leaky. So an example here is the leaky potassium channel. Those which do require receptors, however, have 2 types of conformations. There's open, which means it's active, so things can move through it, and then there's closed, which is inactive, so things can't move through it. This is just a little note about the stress-gated ion channels. I didn't actually know this, but they're actually the auditory hair cells of the corti organ in the cochlea. So just a random fact there. And actually, before I go on to nerve impulses, I just want to put this in a little bit. These types of channels are actually really important for osmotic balance. So because your water can pass through freely across the membrane, we have to use the sodium-potassium pump to maintain the balance of water, so cells don't sort of lysis or like gain too much water or lose too much water. So when we've got water inside of a cell, there's osmotic pressure sort of pushing outwards, and there's molecules on the inside, so you've got a low solute concentration outside the cell but a high solute concentration inside the cell because you've got a lot of molecules inside of it. But then osmosis happens and this water moves into the cell to try and reduce this concentration. So you go sort of from like a shriveled up really salty chip to like a swelled chip because the water's moved to reduce the concentration of the salt that's inside of it. So when we're talking about nerve impulses, we have to mention the fact that there's a voltage difference between the inside and the outside. So this means there's a membrane potential because there's different concentrations of ions on each side. So for it to be a positive, there's a lot of sodium ions present, and for it to be negative, there's a lot of potassium ions and then negatively charged proteins, because you're probably wondering, wait, potassium's got a plus charge, but there's a lot of negatively charged proteins as well, and there's less potassium than there is sodium, so altogether it's a negative charge. This means there's an electrochemical gradient. To start off a nerve impulse, a nerve cell has to be exposed to a stimuli. This stimuli has to be strong enough to cause the voltage-gated sodium ion channels to open. So now these channels are open, you've got sodium ions moving inside across the membrane into the nerve cell or the neuron. This causes the inside to depolarize as it now becomes positively charged instead of negatively charged. The max positive charge that the inside of the neuron can actually reach is around plus 40 millivolts. This is quite a big change actually from the resting potential, which is around minus 70 millivolts, and this peak is called the action potential. Once it reaches this plus 40 millivolts, it causes the sodium channels to close and then the potassium voltage gated channels to open. This means the positively charged potassium ions can then move outside of the neuron and then cause the inside of the neuron to be negatively charged again. This is called repolarization and it drops it back down below the threshold. But because these voltage gated channels are open and you've got the ions moving out, sometimes it happens too fast because of the membrane potential difference, so it can drop a bit below the minus 70 millivolts. This is called hyperpolarization, but it soon levels off again. Now, I mentioned a bit about a neuron just then, so I'll tell you some of the features of it. So you can see here it's sort of got it's a bit like a tree, but not at the same time. So at the top, on the left hand side, you can see there's a cell body with a nucleus inside of it. I'm sorry I've not labelled that. The little branches, I guess you could call them, of the tree. They're called the dendrites. They receive signals from neighbouring neurons. You've then got your long, middly bit, which is called your axon. This can vary in length from 1 millimetre to 1 metre. Now, I have kept this as a basic diagram because I'm also a terrible drawer, but this is often also coated in something called a myelin sheath with Schwann cells. This myelin sheath has little gaps in between each of the Schwann cells called the nodes of Ranvier. It allows for saltatory conduction, which means basically the impulse can travel faster along it. So if you have myelinated neurons, you get a faster transmission of an impulse. So we have a resting membrane potential, which is the minus 70 millivolts. We then have a little stimulus that causes the membrane potential to go a bit above the threshold, which you can see is the minus 55. And a key thing that you need to remember that this is all or nothing. So if it gets to minus 54, then woo, well done, you're going to reach an action potential. If it gets to minus 56, then nope, nothing's going to happen. It has to pass that threshold, otherwise nothing will happen. But when it does, you've got your depolarization as your sodium voltage gated channels open and your sodium ions enter the nerve cell. This causes an increase in the membrane potential up to around about your plus 40, which is the action potential. This then causes the sodium voltage gated ion channels to close and the potassium gated ion channels to open, causing your potassium ions to move into the neuron and make the inside of it more positive. This is called repolarization. But sometimes these ions can move in a bit too quickly and a bit too many. This causes the inside of the membrane to actually drop below minus 70, and this is called hyperpolarization. But eventually it sort of sorts itself out and levels back off again, back to the resting membrane potential of your minus 70. So I mentioned a little bit earlier about the synapse and sort of what goes on here, so I'll cover it for you. So at the end of your neuron, you have the terminal branch, and then at the very end of a terminal branch, you have something called a nerve terminal. It's called a presynaptic nerve terminal because it's before the synapse, so pre-synapse. And here there's a little synaptic vesicle that's full of neurotransmitters. And then in the membrane, there's actually some calcium voltage gated ion channels, but without an impulse, these are closed. But then as soon as a nerve impulse arrives from the axon, these voltage gated calcium ion channels open, allowing calcium ions to move in. This then causes the synaptic vesicle to fuse with the membrane and release the neurotransmitters. These then diffuse across the synaptic cleft, which is the gap between a presynaptic nerve terminal and then the postsynaptic cell, so the neuron that it's sort of connected to and right next to. In these postsynaptic cell dendrites, there's a neurotransmitter receptor that's specific to one type of neurotransmitter, so it may have 3 different neurotransmitter receptors for 3 different neurotransmitters on it if it's got multiple connected neurons to it. And then the same thing has to happen again. The stimulus of these neurotransmitters has to cause it to cross the threshold for an action potential to be generated and the nerve impulse to move down it to the next neuron and then the next one and the next one. Sometimes they go through things called summation, so when there's multiple neurons connecting to it, it needs quite a few neurotransmitters to cause it to cross the threshold. Sometimes it's just one. It all depends on the neuron.